And so Dalton got his bachelor's degree at the University of Kansas. And then he's basically been a PhD student working with Beverly and myself for quite a few years now and doing really exciting work. And he was also a student in the RGNO course himself. And he's going to tell you today about some of the exciting work that he's been doing.
Yeah, thank you for the introduction, Jake. Okay, yeah, so as Jake said, I'm Dalton Lepridge, PhD student here at the University of Minnesota. And I'm gonna follow up on some of the stuff that Jake was talking about and using microscopy to investigate metabolisms in the giant sulfur bacteria, Thiomargarita. Here are my keywords. So we have confocal microscopy, pH imaging, endosymbionts, sulfur oxidation, nitrate respiration, and chemolithotropy. Okay, yeah, so just a little background, and Jake gave some of this, but yeah, we're talking about Thiomargarita. They're the largest known bacterium, some of them reaching up to 1 millimeter in size. As you can see here, compared to this Drosophila fly, they're massive. Yeah, they were first discovered in 1999, sediments of the Benguela upwelling zone. They're chemolithotropes. They get their energy from reduced sulfur sources, such as hydrogen sulfide or elemental sulfur. They can store elemental sulfur and polyphosphate. The vast majority of their cellular volume, as you can see here in this photo, is taken up by a central nitrate vacuole, where this green layer here would be the small cytoplasm. They're non-modal, which could be one reason why they are so good at storing phosphate, sulfur, and nitrate. They also have a large mucus sheath with a whole epibiont population. And as Jake talked about too, they have an endosymbiont population that's related to, hopefully I don't butcher the word, I never get it right, ciboglinid tubeworm symbionts. They have genes for sulfur oxidation, denitrification, and carbon fixation. So some of the questions, though, that we have, I mean, it looks like the cells can be passed from mother to daughter cells, but there could be other routes of infections, such as endocytosis or phagocytosis. And you know, one reason we're kind of thinking about that is like there's parts of chains that we're passing from mother to daughter cell makes sense when the cells are dividing, but there's also instances where you see some cells within a chain are not infected, but the ends of the chain will have a cell that is infected. So how is that actually occurring then? Yeah, so how do they get there? You know, what are they doing, and what are some of their interactions with Thiomargarita? And just to put it in broad context, the reason for studying these endosymbionts is to get a good or better understanding of the endosymbiotic theory, which is the kind of the generally accepted route for the evolution of eukaryotes, where we had the engulfing and the enfolding of the plasma membrane of a protocell, and it took up an aerobic heterotroph, which eventually became the mitochondria, and it engulfed a photosynthetic prokaryote, and that became the chloroplast here. But yeah, how long did that interaction actually take to occur? Because presumably at first this was probably some sort of parasitic relationship or infection, and then over time it became mutual. So Jake talked about this here with using fluorescent D-amino acids, or FDAAs, to label Thiomargarita, and it's labeling their peptidoglycan. And what's really interesting about this is you think about bacteria with a thick peptidoglycan layer and it being rigid. But what this is showing is that the peptidoglycan layer is actually invaginating in, along with the inner membrane, and potentially the outer membrane enfolding too. Some of the open questions here are, what are the vesicles used for? Do they enfold like a phagocytosis-like process? Are they storing nitrate? Yeah, kind of what's the function of that? And that's what I'm working on, more or less, is I'm using confocal microscopy right now to measure the pH in the cells, both the endobionts and inside the vesicles in Thiomargarita. There's a few metabolisms that are relevant to this system that could provide a localized pH change and may give us some hint as to what's going on, such as the DNRA pathway, where you're going to end up getting ammonium, and this would increase your pH here. We could do sulfur oxidation with O2, and that's going to create protons there and give you localized acidity. You could also have a denitrification pathway with elemental sulfur that could lead to acidification also. The vesicles within the genome, it's showing that we have V-type ATPases, which these consume ATP or hydrolyze ATP, and are thought to pump protons into the vacuole. So this is essentially what I'm doing here, and I'll show you some pictures here moving forward, is we're Jake labeled cells with FDAAs, which was showing that peptidoglycan is enfolding into these vesicles. So I was using BODAP 510-512, which is a fluorescent fatty acid, and it's pretty widely used in eukaryotic systems to show membrane trafficking or just a general membrane stain. And I'm using pH Roto Red, which is a fluorescent dye that's used to measure the intracellular pH. And then Hoist, it's just a live DNA stain, staining double-stranded DNA. And this is a confocal image here, about halfway through a cell. So this is actually showing what's on the inside here. And we can see that Thiomargarita's membrane is being labeled here. This is the sheath area surrounding it. And then this is another cell here. Here's a transmitted image. And we're focusing here on this vesicle here, which has endobionts in it. We can see that the green, this is our BODAP label. So this is showing fatty acid incorporation here. This is also co-localizing in this location. And then here is the pH Roto. And how the intensity works with pH Roto is the more intense, the more acidic the solution is. So you can see here in this cytoplasmic region, this is more neutral, whereas in this region, this is going to be much more acidic. And when you overlap them, you have this co-localization between all three channels in this one region, which suggests maybe that there's some proton pumping occurring from the vesicle, or there could be an acidic metabolism coming from the endobionts. And this was kind of early on when I was doing some of this work, and I don't think I was using the BODAP or the fatty acid stain long enough. As you can see, Thiomargarita has a very thick sheath, so that's just a lot of distance to travel through. So when I stained longer, I ended up noticing that not all, but a lot of Thiomargarita cells have this large central vacuole in it. Kind of seen in the first image from the Schultz paper that I showed earlier. So in red, we have pH Roto, and this is just showing the pH here. In this chain, this is about pH 5.5 to 6.5. And green, this is labeling our vacuole here. You can see that we have other smaller vacuoles outside of this within the cytoplasmic region. I know the green is a little overwhelming in the this image here, but there are endobionts in these two labeled vesicles here. These have an even lower pH in this region here. And just for a reference, this is an XY view, and then we just take a slice down the middle and flip it. So this is the YZ view, where you'd just be turning it. And then this one is flipping it up into the XZ view. So you can see that this vacuole is fairly acidic relative to the cytoplasmic region, where it's around pH 7.5. And then this is another cell with about the same situation, where we have an inner vacuole of a pH of about 5.5. There's even some actual vesicles or inner blebbing into this central vacuole here, with a lower pH of about 4 to 4.5. And that goes for the same with these other cytoplasmic vacuoles here. And I think the stage moved when the image was being taken, and that's why I apologize for that here. It seems that there is some proton pumping probably occurring into the central vacuole, and maybe there's some sulfur oxidation occurring from the endobionts that are also leading to acidity. I used the VATPase inhibitor, bifilomycin, to prevent the pumping of protons. I did the same experiments that I just did there. You could call those all control images. And then I added in bifilomycin prior to doing pH staining. And here are some of the results that I'm coming up with now, where I have the central vacuole now is around 7.8, 8.0. These other vacuoles may be right about in the same range here. And then the cytoplasmic region, fairly in the same range, maybe slightly more acidic, around 7.5 in this image here. What's interesting though is that, and Jake kind of touched on this with the TEM images, is that it looks like most of the endobionts are within vesicles. That's not always the case though. It does seem that there's some that are free floating, or they're not showing up with the Bodepi dye, and that they're not membrane bound. And in that case here, where you can see that you have co-localization between this red spot here and the blue here showing the double-stranded DNA, it's showing that it's still acidic in this region, which is about a pH around 5.0. This is another one with bifilomycin added in this case. And again, the central vacuole is around pH 8. The cytoplasmic region is slightly more acidic, but right around the same. And then, yeah, we have these regions here where you have this co-localization just between the blue and the red channels, which would be the DNA and the pH roto, which are still fairly acidic at pH 5 to 5.5. So while the vacuole is not acidic, nor the cytoplasm, it seems that some of these free living endobionts are still acidic. So maybe they are still doing some sulfur oxidation there, and VATPases are inhibited here, which is preventing proton pumping into the central vacuole. And this is just a really cool three-dimensional image of this previous slide. So if you just cut the half, I just thought it was a really good way to visualize this vacuole. And you can see some of these other smaller vacuoles too, outside of this large one here. And then you can see there's additional blebbings into the actual central vacuole too. So it does seem that some of these vesicles are fusing into the large central vacuole there. And that's what I have for now. I do have some other experiments that I ran. I'm still going through some of the images with them though, where we've been staining for nitrate reduction intermediates, so staining for nitric acid or nitric oxide. I'm sorry, and using some other inhibitors, such as inhibiting the electron transport chain, or F-type ATPases. But yeah, in summary, I have seems mostly endobionts are confined to vesicles. And for the most part, vesicles are acidic, and that vacuole acidity seems to be in part coming from the ATPases. And that's what I have. And lastly, I just wanted to thank Kurt and Jake for putting on the RGNO field course, and inviting me to give a talk here. I was a first generation college student. Didn't really think I was even going to do undergrad. And then I did that. And then I ended up getting into a PhD program with Jake, and never in my wildest dreams would I have thought that I was going to go to Africa and do research. So I just wanted to show a couple of pictures, if we have time, to say my thanks and some of the fun stuff that we did there. So here was Jake picking Thiomargarita cells on the ship. Probably not his favorite thing to do. He gets a little motion sickness, and then you're under the oculars and it makes it 1,000 times worse. That was fun. And then we had, here's us grabbing the core and pulling Thiomargarita sediments out of there, or about to. Here was Martin demonstrating how to use the flotation devices for everybody. There was the mirabilis. That was a great time. Here's Kurt. He was showing me how to use the fluorescent microscope over there, where we initially started doing some of this imaging with some different dyes, but this is where it all kind of started, was measuring these the acidity within Thiomargarita all originated here. Here's me and Nathan and Martin. We went hiking out at the sand dunes one day. That was an incredible day. And then we also went to these lichen fields where it seems like miles and miles of just nonstop lichens. That was a pretty amazing day too. So that's what I have. Thank you, Jake, Beverly, Ryan, Nick, the Mirabilis crew, and my funders, NSF and Simons Foundation.